Morning, students. Today we will study the difference between the F transfer and HFR transfer in bacteria. Following are the notable difference between the F transfer and HFR transfer. It takes 95 minutes for an entire bacterial chromosome to be transferred, in contrast with about 2 minutes for the transfer of the F. The difference in the time is the result of the relative size of the F and HFR chromosome. During transfer of HFR DNA to the F minus cell, the mating pair usually break apart before the cells separate. In a mating between HFR and the F minus cell, the F minus recipient usually remains F minus because the cell separation almost always occurs before the final segmentation of F is transferred. In HFR transfer, the transferred DNA fragment combines with the chromosome of the recipient, thereby generating recombination of the F minus cell. Difference between the conjugation and transformation. Conjugations differ in the two ways. Conjugations require direct cell to cell contact. The conjugating cell most generally be of the opposing mating type. Donor cell must carry the capital F factor and the recipient cell usually do not. Genes transfer by the F dash factor sex deduction and HFR cell is produced when F factor integrates stably into the host chromosome. The only occasionally the integrate F factor requires from the chromosome, and the HFR cell revert to F plus cell. In doing so, the F factor carries along with it some chromosomal genes that were adjacent to the F and becomes once again a cytoplasmic, autonomously replacing the particles. The exercise F particle is now called the F dash, and the F plus is called the F prime cell. The F dash cell behave like a F plus cell, and during the conjugation between the F dash cell and the F minus cell, F plus particle or the sex factor together with its attached chromosome gene is quickly transformed to the F minus recipient. The recipient cell then become a secondary F dash cell. It is a partial diploid for those genes it received from the primary F dash cell. Recombination of this type is mediated by F dash factor, and it is called as the sex deduction or F deduction factor. Plasmids and episomes. In addition to the normal DNA chromosome, extra chromosomal genetic materials or the elements are often found in bacteria. These elements are called plasmids. They replicate independently of the bacterial chromosome. Most plasmids remain autonomous in the cytoplasm. Some plasmids are capable of either replicating autonomously or integrating into the bacterial chromosome and are called episomes. Plasmids are circular piece of supercoiled DNA about 1 by 100 the size of the main chromosome. They are found mainly in almost all strains of bacteria tested, but also in some eukaryotic microorganisms such as Saccharomyces cerevisiae. One of the most important characteristics is that they are not essential for the normal activities of the cell in which they reside. In many cases, some genes carried by the plasmid may be useful to the cell in certain circumstances. Following are the useful character. They provide the bacteria with the ability to conjugate by the formation of F. pili, the ability to become the resistant to various antibiotics, the ability to make a toxin, toxigenicity, or the ability to use the unusual compound as a nutrient. Although some of the plasmids may be identified with a particular functions, may have no role and are called as cryptic plasmid. Thank you.